Okay, good morning, everybody.
Good morning.
Today, we are going to talk about enzymes, enzymes and the mechanism of enzymes. We normally call it usually as a lock and key model. And there are some inhibitors also, they affect negatively the process of enzymes. But before I start, I would like to mention something about enzymes. Like, what are those enzymes? They are actually helps us to take process of biological, as you said, biological machineries or cellular machineries, that they are the ones that are having the process, helping the process of laboring in our metabolism, in our body, actually. Before we proceed, what is metabolism? Metabolism is all the chemical reactions that takes place in an organism or in a body. Okay. So, as you all know, we are always having chemical reactions, or actually all organisms, all living organisms, they are having chemical reactions non-stop throughout. Okay, throughout chemical reactions. Can I give you an example for this? For example, we always talk about food particles or nutrients are broken down by oxygen to release energy, isn't it? The process is called as respiration. So this is done by enzymes. Guys, normally if we leave it to nature, its own entropy, food molecules will be broken down by oxygen maybe once in a year or more than that. If we leave it, even it won't be, it might not be take place. Okay. But enzymes, they come and they work like cellular machineries. They come get the food molecules and oxygens together and break that. Like they use oxygen like a hammer. While chemical bonds are broken down, energy released. So this process is one of the examples that's how enzymes are important. Even DNA replication, it is done by enzymes. And also we said like carbohydrates, like monosaccharides, disaccharides, that's condensation process, or opposite of condensation, hydrolysis. Those are also done by, both of them done by enzymes. Okay. So what is the mechanism of enzymes, and why do we call them as a lock and key model? Guys, each enzyme is having its own unique way. Okay, this is if this is our substrate. Substrate, we are going to call it a substrate. It might be either food molecule or something else that enzymes are working on it. They are acting on it. Okay. Enzyme is, we are going to use this key as a representative of enzyme, and it has its own specific way. You know, I can't open other padlocks that someone else's padlock with the same key. Okay, that's why we call enzymes specific to substrate. So for this padlock, I can only use this key. If I have another padlock, it is not going to be right with my key, is it? So that is also another important and unique thing about enzymes. They are specific to specific nutrients only. Like, for example, sucrose. Okay, sucrose is a monosaccharide or disaccharide? It's a disaccharide. Sucrose is having enzyme. Let us call it sucrase. Okay. It can only react with that enzyme, not oxidase or not amylase or not another one, but sucrose to sucrase. Okay. Yani one specific enzyme for a specific substrate. Okay, that is another important thing. And then we also are going to mention about inhibitors. Inhibitors, that some chemicals, they prevent enzymes, like they act like enzymes, like fake model of enzymes, and they prevent or stop or block the enzyme to work on the substrate. Those are handicaps for the chemical reactions. Any questions so far? No question. Okay, let's also illustrate a bit more about the mechanism. This is if, let's say this is my substrate, okay. Or let's say this is my enzyme. Okay, this is my enzyme with a specific shape. You can do any shape. Okay. I just choose randomly this one. And this enzyme needs a substrate or food molecule, food or nutrients that react on it, and then it's either going to combine them to a complex molecule, into a complex molecule, or divide them into a simpler molecule. Okay. And let's assume that the substrate is something like that, so that they can match, is it? So they can match like that. And this guy is called, we're going to call it as a substrate. Remember from the question that I gave you yesterday. There was a substrate question. So they are going to have, enzyme is going to react on the substrate, enzyme is going to react on the substrate, and we are going to have something like that. Something like that. Okay, so they match, they fit within each other. And now this combination, this is called as an enzyme, substrate, what? Enzyme substrate complex. That is going to be called as an enzyme substrate complex. And the product, okay, and the product will be what? Product enzyme, do you believe or do you think that enzyme, the structure of enzyme is going to change? Huh? It will change. Okay. It will not change actually. Okay, the structure of enzyme will not change because the structure of enzyme change at the end of the reaction. This enzyme means we can't use it anymore. If it is, there's another thing now you like now. The structure of enzyme, even after the reaction with the substrate, no change. Okay. The structure of enzyme has no change. If it's changed, then this substrate always will remain a site. Okay. Since they are specific to this substrate, if the structure of enzyme, actually this is done by inhibitors. They change the structure of the enzyme, that they cannot match with their own substrate, then this enzyme is not. Understand? So they have to maintain their structure to find a similar nutrients and to go ahead. Okay. Thank you, Iman, for bringing that information. The chemical reaction, the chemical structure has no change even after interaction engaging with its own substrate. Okay. So our enzyme is still the same guy, but now our substrate. Let's say this is our maltose. Okay. Let's say this one was maltose. Maltose. What is the building unit? We want to break it down. What is the building unit of maltose? Glucose plus glucose plus glucose. Our product, enzyme and our product. Our product will be, if this one was maltose, this will be glucose, glucose. Okay, so this is our product, and this is our enzyme. Okay. Very simple mechanism. It just looks like kilo model. Enzyme act on a complex substrate and they form a complex enzyme substrate complex. And at the end of the day, we will find our enzyme just the way it is. And the product now is different. That is the only difference. That is the only change. Okay. It can, how about the opposite way? Is it possible? Opposite way. Yes, it is also possible. It can get two glucose and combine them and get maltose again. So it is reversed. And this is another properties and functions of enzymes that they are having reverse reaction. They can either build up a bigger molecule while they can break down a small molecule. Okay. So that was a look and key model and mechanism. Is there any questions so far? Is there anything blurry so far? Clear? Okay, good. Now let us take note some points. After this mechanism, if we have drawn it, okay, 1 minute to draw this mechanism with its labeling. It's important. Enzymes, substrate, food. You can also remember it as a food, but let us use it as a substrate. You can remember it as a food or nutrient, but officially they are substrate. Okay, let us get used to this kind of stuff. This one, this is called from. Actually, this is the same thing. You know, we used to call it like that. Maltose, equal glucose plus glucose. Remember, so now we are we building into a complex molecule or we are breaking down it? We are breaking down to simple glucose. So the arrow is this way. What is it called? Hydrolysis. Exactly. It can also either be this way. You know, enzymes, by the way, enzymes, guys, it can be like that or like that. Have you seen that one? It means this process goes reverse. It can either go to right-hand side or left-hand side. Okay, 1 minute for you to complete drawings. Okay, you're writing? Okay. Right. Labeling is important, guys. We do it exam with substrate. You might be asked during exam time, you might be asked like illustrate the mechanism of enzyme or illustrate the lock and key model of enzyme. So labeling is important. Yes. That is actually the chemical reasons they are using this change. It is not the issue at all. Maltose. You know, it was my job and only Mohammed is aware that there is a missing element. Oh, it was my second job, sorry. Okay, are you ready? Okay, 10 more seconds, and we have little explanation that need to be taken down. Okay. Number one, substrate combined with the active sites. We are taking notes now, like step by step, like a list of mechanism. So we will remember tomorrow and later on. Number one, we explain them one by one. Substrate combines with the active site of the enzyme. Substrate combines with the active site of the enzyme. Together. Yeah. Okay. By the way, I think you might be having a perfect question. What is active site now? We never mentioned. Any idea? Maybe you go through it before. Active site, guys. Active site of an enzyme. Hisham, where it would be? Where it might be. If you would like to put active site somewhere on this enzyme, where would you put it? Here. Here. Okay, that is 11 out of 12. You get 11 out of 12. So we can specifically, where is it? Not the space. It's not the space. Yes. This document. This document. Actually, also what we can say is the parts that touches the parts that touches the substrate. Okay, the parts in enzyme. This part. Active sites. Okay. Because you need to take note this one. Mushtan, you need to take note this one because this usually comes in exam. Active sites, that is the part that is able to interact with the substrate. Active sites. Which is active, you know, which is going to react on the active site. This one, this part, you know, we said substrate combines with the active site of the enzyme. Now, that one is active. 2. Active site is induced to change shape slightly by the substrate. Active site is induced to change shape. Like it's like hugging each other. Yes. Slightly by substrates. It's kind of like hugging each other. They like engaging and combining. In a bracket, bonds are broken or build up. The bonds, chemical bonds, are broken or build up. Isaac, you good? Yes, this is number 2. No, continuation of number 2. Chemical bonds are built or broken. Number 3, substrate, enzyme. Substrate and enzyme forms enzyme substrate complex, or they form enzyme substrate complex. You know, they induce each other, and then they form complex, and then product. Number 4, products leave active sites. Products are going to leave active sites. Finally, number 5, SITE. Yes, TE. Number 5, active site returns to original shape. So it remains its own original pattern. Continuation. Let me finish. Continuation. Now they can accept new substrate molecule, or now enzyme can accept new substrate molecule. Returns to original shape. Huh? Where? Now enzymes can accept new substrate molecule. Okay. Now let's actually we already summarized the properties of enzymes, but it's also good to take notes about the properties. Yeah. So far, any question? Any question? Yes, Isaac. Exactly. Or because since we said there are reverse reactions, they can either break it down or build it up. Okay, it is reverse reaction. Enzymes are able and capable to have reverse reaction. They can either, remember, we said maltose. They can either combine glucose and fructose into sucrose, or glucose plus glucose into maltose, or they can break maltose into glucose. Yes. There are so many types of enzymes because they're unique, like you, like human beings, they're unique that we can't compare them with another enzyme. So for every single molecule, like glucose has own enzyme, sucrose has own enzyme, starch has own enzyme. Okay, oxidation, hydrolysis, condensation, they all having its own unique enzyme. Just unique, just like you are, guys. Okay. Now, properties. Properties of enzymes. Let us write a bit more about properties of enzymes. First one, number one, they are protein in nature. So if something affects proteins, enzymes also will be affected. They are protein in nature. Like we are humanists in our nature, isn't it? Okay. Number two, they are sensitive to temperature and pH. Thank you. Number three, they are reusable. They are reusable. You can see here, guys, we already done with maltose, now it's ready for another molecule, is it? So they are reusable. Was there any problem? Number four, they are substrate, what? Substrate specific. They are substrate specific. What is, by the way, who can remind us what does it mean, substrate specific? Yes? Exactly, they're unique, isn't it? Particular enzyme affect particular substrate. So we call that phenomena as a substrate specific. Number 7. Okay, thank you for correction. Number 5, enzyme catalyzed reactions are always reversible. Enzyme catalyzed reactions. Okay. Enzyme catalyzed reactions are always what? Reversible. What does it mean? It is reusable. You're talking about reusable. Reusable. They're reusable. They can start with once they are done with a substrate, they can get another one. So they're reusable. We can use enzymes again and again. Okay, who is absent? Which one? Reverse or reusable? Reversible. Yes. Reversible, what is the symbol of reversible reaction? One arrow, half arrow, another half arrow, isn't it? Imran, do you remember this one? Reversible, it can either go this way, builds to bigger molecule, or can go this way, or like opposite way, or contrary way, to smaller simplifier molecule. Imran, okay? So they are reversible. This also symbol can remind you, and you will see this in the question. You will be asked, you will be given this icon, blah blah blah, and then that icon, the product, you will be asked, what reaction is it? You need to know that this is an enzymatic reaction because it's reversible. Okay. Reversible is like that, and reusable is again. What does it mean reusable? Caleb. Again, isn't it? You see here it's already done. Now what is going to hunt another, going to hunt another substrate. And they, okay, finally, it is number 6. They have a particular shape. Enzymes having particular shape. Anyone missing any points of properties of enzymes? Yes, they are very efficient, but we can use this in as functions, huh? Or properties. Okay, let it add as properties. They are very efficient, so little quantity will be done. They are very efficient, even little quantities will be done. Okay, done. Good. Wow. Now, some people have this saying called lactose intolerance, which means that enzymes are toxins. Which means in the body, lactose intolerant people they have that enzyme to break down lactose. Understand specific enzymes. Thank you very much. It's an example. You killed it, fella. Now, okay, another type of title, functions of enzymes. Functions of enzymes. Guys, because of the compact syllabus, we have to put it. Number 1, there are 2 functions usually, and it usually comes an example. What is the key? Number 1. Enzymes speed up metabolic reactions. That was our introduction. And I think speed up metabolic reactions that otherwise it would be too slow to sustain life. Actually, impossible. Even not even too slow, but impossible. But okay, let's give it like that. It would be too slow to sustain life. Okay. Actually, by random, I can't give it random. Food specific particles are going to be broken by oxygen. Energy, blah blah blah, guys. This is not, it can't be given to us randomness. Not too slow, actually impossible, but since our curriculum says this way, we use it. Number 2, specific in nature, comma, An enzyme will only act on a particular substrate. Particular substrate. For example, this is important. Sucrase, guys, we write like that. Okay. We will come to it, but let's use this table. Sucrase acts on a sucrose. Okay. Sucrase. Be careful, spelling is different than sucrose. Spelling is slightly different than sucrose. Sucrase acts on a sucrose. Okay. So they also, as we said, they are substrate specific. They're unique according to their substrate. Even their names are related. Enzyme, name of the enzyme and name of substrate also familiar. Okay. Any questions so far? No question? Yes, Kiran. Burst or reversed? How is it reversed? Oh yeah, good question, guys. Thank you. Guys, you know, we say sucrose can only act on a, say sucrase, can only act on a sucrose, isn't it? The limit is here, sucrose, fructose and glucose, isn't it? So this enzyme can only recognize glucose plus fructose, isn't it? And it goes, let us use one arrow. Normally enzyme should be double arrow like that. Enzyme should be indicated like that. Okay. Now, glucose plus fructose, what might be my product? Sucrose, isn't it? So, what enzyme here I'm using? Sucrase, okay? Okay, what enzyme? Sucrase. Now, let's talk about another chemical reaction. It doesn't mean that it is bending. Understand? It keeps building, then it stops and it comes back to break down. It's not like that. Okay. It is not like continuation, breaking down, building up. Now it is getting sucrose, but it has to be sucrase again because it can recognize all of these guys. Okay. Now sucrose is going this way now. Or let us let it be now. Now I have a sucrose, it's going this way now. And I need the same enzyme to break this sucrose now. Actually, it depends on the inquiry of the organism. If organism says, you can get some water. If organism requires, actually, Kiran, different styles like that. If organism requires more sucrose, then enzyme will come find glucose and fructose to form sucrose. Okay, another day, metabolism or organism requires more glucose and fructose, and enzyme is going to find the sucrose and break it down. That is the way actually we call it reverse. Understand? They are not like confused, like building up and it stops and comes back, breaks down. It's not kind of such thing. It builds up them all until organism is saturated, until organism says, okay, I don't need anymore. And then another day, body is going to call enzymes that now I need you to break down sucrose. Okay, then they come and they break down. That is what we mean by reversing. Okay. It's a reversed process. Reversible. Oh, the process, it might be like either hydrolysis or condensation. It depends. Or oxidation. Oxidation is broken down by